The title of this presentation is Extensions of a Recent Fast Quantum Algorithm for Structure Prediction of the COVID-19 Receptor. To begin with the presentation overview, we'll be introducing fast quantum algorithms and being able to relate fast quantum algorithms to other types of quantum algorithms and quantum computing in general. And in doing so, we'll be drawing upon previously examined enzyme design frameworks from some other type of experiences and other types of compounds I've looked at from a couple of other videos that have been posted on this channel. And we'll be looking to highlight a few compounds that can be analyzed in the current near-term hardware with quantum computing, as well as also discussing characteristics of computations on the quantum side that are difficult to consider in the near term and what the type of difficulties exist, but still in the current near-term hardware, how we can take advantage of our current knowledge to at least make some hopeful progress. So the references that we're going to be including and looking at is the first one, which is going to be the main focus, which is titled A Fast Quantum Algorithm for Protein Structure Prediction in Hydrophobic Hydrophilic Model, which is by Wang and Cheng, as well as a second reference for Variational Quantum Algorithms for Nonlinear Problems by Lubash et al., as well as a third final reference for Quantum Algorithms for Electronic Structure Calculations, Particle Hole Hamiltonian and Optimized Wave Function Expansion by a large group of authors. So we're going to be looking to relate all of these papers together from the fast quantum algorithm perspective and to discuss how this relates with certain topics of interest such as metabolic engineering, enzyme design, or directed evolution of enzymes. And we'll first have to introduce a couple of objects which are discussing the things that we'll be needing and how this will demonstrate the connection between molecular docking and looking at enzyme design and directed evolution of enzymes and making a concrete connection with quantum computing. And in quantum computing, we have to keep in mind that we usually have to have some initial encoding of the initial data of the problem with the ansatz. And the ansatz is comprised of quantum circuits, which can include C-naught gates, Toffoli gates, or quantum Fourier transformation gates, as well as other gates, which I've indicated here, which are given by a hat of a poly Z rotation, which signifies taking a readout or a measurement from the quantum circuit that we're time evolving to obtain some information from, as well as input and output ports into different parts of the circuit, as was described in a Variational Quantum Algorithms for Nonlinear Problems paper by Lubash et al., in which they look to be able to leverage quantum computing to solve highly nonlinear problems on differential equations, including the Burgers and nonlinear Schrodinger's equation. For our purposes, we'll also be able to make use of some properties of a Hamiltonian and time evolution for the quantum chemistry type problem and molecular docking framework problem that we're going to be introducing. Besides the pieces or the components of the quantum circuit that we have to look to encode for the initial encoding of the ansatz for a quantum computing problem, we can also look to have a probability measure, a probability amplitude on k qubits, and a qubit is given by a fundamental unit of quantum information, which are initialized to 0 in a quantum circuit in which our time evolved forward. So in this case, we're encoding a probability amplitude for some quantum state psi, which lives in the Hilbert space, and looking at a decomposition of this quantum state into a probability amplitude in which each one of the terms which are given in the summation of the index for I encode the probability of the state living in this state ket of I. And besides the quantum circuit components and the probability amplitude encoding, the chemistry and the molecular docking and the enzyme design framework comes into play because we can define an electronic structure Hamiltonian, which is given by a type of Hamiltonian, which is helpful for chemists and material scientists and other types of people in those adjacent fields for looking at different electronic structure and properties of materials that can be studied. And in this case, the Hamiltonian for the electronic structure is given by the fact that it can be expressed in terms of a product of annihilation and creation operators, which is given in the first summation of I and J, where we have a hat of I the Hermitian conjugate, which is the annihilation operator, whereas the other operator, which is given right next to it, which is hat of AJ, is a creation operator, as well as another term, which is a sum over I, J, K, and L for a hat I J, or a hat of AI for the annihilation operator, as well as hat of AJ for the annihilation operator, as well as two creation operators at the positions L and K. And from the Hamiltonian, from the electronic structure, there can be a lot of connections with statistical mechanics and thermodynamics, because we're looking to study which compounds can form a stable resulting complex after the binding and chemical reaction has occurred. And to this end, we can make use of the Hamiltonian, which can be used in some other cases, more theoretical cases, to describe to encode a probability measure. But in this case, we can look to describe a 1-point and a 2-point electronic correlator, which is given by an expectation value as shown on the slide from these two expectation values, in which the 1-point electronic correlator is given by the fact that we only have I and J in the outermost bra and ket terms for the quantum state for the expectation value that we're looking to approximate with this integral with respect to the variable dr1, as well as from the fact that the 2-point electronic correlator is given by a very similar expectation value, with the difference that we have ij instead of I, as well as kl instead of j in the outer in the outer and innermost terms of the expectation value. In addition to the fact that in terms of the integral, we also have another variable dr2, which we're integrating against for this function that we're trying to approximate the expectation value of. And I had mentioned in the previous slide that there are several types of components that have been looked into previous work, which was looking at more of a numerical perspective for being able to solve partial differential equations with a variational quantum algorithm, and how the quantum circuits which are used for that variational quantum algorithm have a lot of connections with the type of quantum circuits and perspective of the fast quantum algorithm, and being able to use the fast quantum algorithm to discuss what types of compounds could be simulated on current near-term hardware quantum computers, and whether and also what would be the type of breaking point beyond which the current near-term hardware of quantum hardware would no longer be able to effectively simulate properties of these compounds, and how we could maybe look forward to future near-term quantum hardware that could help us simulate more complicated electronic structure. And to this end, running the molecular docking workflow with quantum computing is possible by making use of connections between a fast quantum algorithm that we're going to be discussing more, as well as a variational quantum algorithm that we have been discussing the quantum circuit construction for. And for the variational quantum algorithm, just very briefly to describe a workflow so that we can establish so that we can further build on and establish more similarities that are used for the fast quantum algorithm, is that we start with a nonlinearity space of around 10 partial differential equations, ranging from partial differential equations describing fluid flow, gravitation, electricity, and magnetism, as well as wave propagation. And then we impose some requirements for the ansatz, which is the initial state of the solution that we're using for the partial differential equation, which we would like to time evolve forward across some fixed number of iterations of an optimization loop to determine how well the solution approximation would actually compare to a classically realized solution for some partial differential equation of interest. And to this end, we impose conditions on the Hilbert space before initiating the time evolution, as well as potentially any other initial conditions on the first and higher order derivatives of the solution state. And once we've looked at the nonlinearity space and the types of solutions the types of properties of the initial state of the solution that we would like to have, we can look at formulating a cost function in which the cost function ground state space is comprised of transforming a given partial differential equation of interest into adder and adder-dagger operators, depending on the first, second, or third derivatives, or however high order derivatives appear in the partial differential equation of interest, from which we can form a resulting quantum superposition for the solution state approximation. And finally, we pass to the last stage of the workflow, which is quantum circuit automation with with an open source package from Google Quantum AI, which is CIRC, which allows us to assemble solution approximations for all these PDEs and to determine whether a variational quantum algorithm that was initially proposed on 2 or 3 qubits for the nonlinear Schrodinger and the Witze-Burgers equation can also be extended to much more complicated settings for more complicated partial differential equations, which have many more nonlinearities at play. And in comparison to the quantum circuits used in the variational quantum algorithm paper that we had just been describing in a little bit more depth, the workflow in the previous slide, the quantum circuits for the fast quantum algorithm have a different component which poses experimental difficulties for near-term hardware, and that is related to the oracle gate. So the oracle gate is a component of the quantum circuit that's given in this portion of the circuit that's given here from the Wang and Cheng paper in figure 6. And it's demonstrating how even though there can be some similar use in the quantum circuits of having to use qubits and timing evolving them from some favorable initial state in the Hilbert space, there can be a lot of potential experimental difficulties that can come up from either an oracle gate or different types of operations which are given in their quantum circuit architecture. In this case, the quantum circuit architecture for the fast quantum algorithm in comparison to the quantum circuit architecture for the VQA is a lot different because they describe a couple of qubits which are initially which are designated in these initial states, which are given in the leftmost side of the circuit. And we would like to time evolve these qubits from the initial state under different gates, whether it be a Hadamard test or other types of operations to obtain a desirable or a favorable readout measurement that can be used to make some type of interpretation about molecular dynamics, quantum chemistry, and electronic structure. And in this case, the oracle comes into play because it's one gate that's used that's used for 3 registers in the quantum circuit, and it appears around the middle of the quantum circuit, around the middle of the box portion, which is iterated some like integer number of times, as they say in their paper, pi over 4 square root of n over m times. Which is relating to the coordinate calculation, the energy calculation, inverting an energy calculation, as well as inverting a coordinate calculation, and finally performing a diffusion operation. So it's demonstrating that even though there can be some similarities, roughly speaking, between quantum circuits that are used amongst different types of quantum algorithms, there can be a lot of or many potential difficulties that can arise from different types of gates, which can be used to process the information in the quantum algorithm. And in this case, relating to information of the electronic structure of different compounds. And there are additional points of consideration because even though an oracle or a quantum circuit with an oracle gate maybe could be experimentally realized in the future, in current near-term hardware, it's very difficult to realize all possible quantum circuits that can be written down very easily on paper to actually simulate these circuits and to generate some meaningful type of measurement, which has very much been the efforts of industry leaders such as Google Quantum AI, in which from the image that's given of their near-term quantum hardware with their Sycamore processor on the left, that there are different types of dilution refrigerators which will connect the whole apparatus together and maintain a very low temperature so that the pristine nature of the quantum information will not be lost throughout the time evolution. And the reference that I've given on the right, as I've discussed in another presentation, is very important because it also relates to stabilization of entangled states, which is very much relevant for future near-term quantum hardware, because if we are going to have a near-term quantum device with millions or hundreds of thousands of physical qubits, and let alone logical qubits, we have to be able to look at stabilizing some highly entangled state and being able to time evolve this highly entangled state effectively for generating some type of readout from a state of interest. And so we want to think that if we're talking about quantum algorithm in an electronic structure, Hamiltonian and quantum chemistry, what would be the requirements for different compounds? And because we're in the near term right now, with our quantum hardware not being able to look at all compounds of interest, we basically need to look at a sweet spot of compounds, namely compounds that are not too long, but at the same time not too short, so that the compounds that are not too short would not be trivial for a classical algorithm to approximate the binding affinity of. And this is relating to some previous use of classical computation methods that I've used for molecular docking simulations and approximating binding affinity, because there's an open source repository maintained by a faculty in the Chinese Academy of Sciences, which is called CellTalkDB, which is relating to providing a list and very extensive information from hand-picked receptors and ligands, which can be used to model several different phenomena, whether it be respiratory diseases or other types of diseases in which a virus stages some initial replication phase in a host organism, after which it propagates its genetic material within the host organism to begin an infection. So you can take different types of compounds and run through some molecular docking workflow, different homology models, and pass on different parameters to molecular docking algorithms, including the size of the search region that you want to run the molecular docking simulation on, in terms of so that you can have some idea about possible conformations, which could exhibit different ways in which a ligand and receptor could interact with one another, depending upon the structure of the receptor of interest. And unfortunately, poorly specified molecular coordinates for the molecular docking simulation can result in conformations whose orientations are likely not correct. And this has happened in several different types of instances in which you have to make use of different parameters that are passed to the molecular docking workflow. And from some of the open source molecular docking workflows, which I've used, like AutoDocVina, you have to define the search volume about the space between the ligand and the receptor in angstroms cubed. And then after you define the search region, the algorithm will be able to generate some prediction of the binding affinity between these two compounds by detecting the total number of CPUs, in this case 20 on the specific hardware that I was running, as well as reading the input and defining a scoring function so that it can systematically rank the effective binding affinity of different compounds and different conformations against one another. And after it analyzes the binding site with the help of the scoring function, it's able to perform a search based on the total size of the search space volume for generating an approximation to the binding affinity. And the fast quantum algorithm from the first reference that we provided, it has very much an interesting connection with molecular docking workflows and even computational or classical computational methods for approximating a binding affinity, because it provides a specific characteristics on the scaling of quantum circuits to be time evolved and optimized for generating readout for the binding affinity of different compounds. In particular, the two authors, Wang and Cheng, of that paper, they provided with a polynomial expression in terms of the number of amino acids for the expected number of qubits that would be required in our quantum circuit to perform time evolution. So it's basically saying that there exists a polynomial scaling relation between the number of amino acids on a compound of interest and the number of qubits to be time evolved in the quantum circuit. And in this case, the polynomial, which describes the scaling relation between the number of amino acids in the molecular structure that we're looking at on one side, and on the other hand, the number of qubits in the quantum circuit that we want to time evolve is given by the polynomial 16n cubed minus 24n squared minus 12n plus 63 qubits. And the fast quantum algorithm also takes into consideration nearest neighbor interactions. And this is very much reminiscent of different areas of math and physics in which people like to study and prove statements or look at qualitative behavior of polymers, in which there are interactions between nearest neighbor sites of polymers or even of spin systems. And in this case, In table 1 of their paper, Wang and Chain also also direct our attention towards the fact that there are transitions between adjacent amino acids in which you can look at transitioning from the D-1th site to the D site of the amino acid of a chain of amino acids, and they provide directions and a prescription on how we should actually move in the lattice depending upon which site we're on, as well as the value of these two states, the weights at D of 2 and the weights at D of 1. And from classical computational methods, previous studies have classified characteristics of early variants of the COVID-19 receptor, but such studies for newer variants are not yet complete. And this was very much relating to some simulations that I ran in the classical realm previously, because I was interested in looking at how to make use of a point of comparison between being able to compare a machine learning workflow with a deep learning workflow to determine and to ask whether there would be any substantial difference or drop or change in performance between the machine learning workflow and the deep learning workflow, depending upon the structure and the information of the structure of the COVID-19 receptor that we pass to the algorithm. So this archive paper that I talk more about in another presentation on the channel on protease inhibition, it talks about protease inhibition from 92 crystal structures of SARS-CoV-2, and it's making connections and very much drawing our attention towards to ways in which an initial replication stage for a virus can occur, and how this virus can propagate and infect a host organism and spread its genetic material depending upon an initial binding event of a receptor, as in this case COVID-19, and a ligand or another body or another vessel, molecular vessel within the human body that it can infect. And when I was looking at the COVID-19 receptor and trying to make a comparison between the information that was listed from the deep learning workflow and a machine learning workflow, we can see that there's publicly available information in the PDB repository for the COVID receptor. And this is given by the fact that you have to specify as input arguments to a machine learning workflow for approximating the binding affinity, the ligand and receptor files, and these files have to be in a PDB QT format. And in this case, the online repository for the protein data bank, it provides us with a PDB file for the ligand and receptor for COVID-19 so that we can run some simulations under a well-specified search volume, like how we demonstrated AutoDocVina takes for determining what would the approximate binding affinity be. And we can apply the hydrophobic hydrophilic framework from the fast quantum algorithm to the COVID receptor, because we have some idea about the distribution of the total number of pockets among the surface of the receptor that we could look to or hope to have some ligand or some other molecular vessel interact with. And at each one of these pockets, there can exist differences in the approximated binding affinity that's output by a machine learning algorithm or a deep learning algorithm, like the deep learning framework that I've been describing from the archive preprint from the 92 crystal structures, because there can be different types of PDB IDs that's given in table 2 of their paper, in which they list different types of candidates for receptors, as well as different type of ligand, or for ligands, as well as different type of ligand IDs for each one of the PDB IDs, and how these would fit and interact optimally with different protein with different binding pockets on the surface of the COVID-19 receptor. And this is also not to mention that besides the PDB ID and the ligand ID, there are also various other chemical structures that are given for these compounds when we're looking at in terms of either their hydrogen bonds or covalent bonds. And the additional characteristics of the COVID receptor also related to the fact that there are different types of residues, and if you go around the total surface of the receptor of SARS-CoV-2, we can form a histogram of the amino acids and determine which is the most prevalent amino acid throughout the whole receptor structure, and how this would inform our computations and approximations of binding affinity with either classical methods or quantum computing methods. And this is, as we can see from the histogram plot, that the majority of the residues are GLY 143, as well as cysteine cis 145, respectively at 71% and 43.5%. And now we can actually talk about some types of requirements for quantum circuits in the hydrophobic hydrophilic model and how this relates to being able to potentially use the fast quantum algorithm for examining smaller isolated segments of the receptor, the COVID-19, and how this can potentially be scaled up in the future with more heavy duty and more advanced near-term quantum hardware. And this is given by the fact that we can recall that the authors of the paper, the fast quantum algorithm, they provided us with a scaling type relation for the total number of qubits that we would need in our quantum circuit, depending upon the total number of amino acids are given in the molecular structure. In this case, we reiterate that the polynomial is given by 16n cubed minus 24n squared minus 12n plus 63. And we can see that if we take this polynomial and plug in different types of number of amino acids for compounds that we could be looking at, we can have an idea about the size and the complexity of the different quantum circuits that we would need to be looking at for running the fast quantum algorithm on, or a proof of concept of the fast quantum algorithm on near-term hardware, and whether we could actually look at total, the entire surface of the receptor of a structure of interest, or whether at the current, with our current near-term hardware, whether we would be restricted to only looking at certain patches or certain smaller isolated topologies of the receptor. And in the case for COVID-19, unfortunately, it seems that in the current near-term quantum hardware, we're more in terms of not being able to look at the totality of the receptor at once, because we can even look at for compounds that are around approximately 50 amino acids long from the scaling relation, which is given from the number of qubits that we would need in a quantum circuit from the number of amino acids in the compound of interest. We would see that we would need around 2 million qubits on our near-term quantum device, which is even beyond the current efforts of Google Quantum AI to reach a near-term quantum device with approximately 1 million logical qubits, or 1 million physical qubits, let alone the more difficult goal of being able to encode 1 million logical qubits effectively. However, we can see that as a starting point, we can look at smaller, less developed and less complicated compounds that are approximately around 10 amino acids long, in which case we would need around 14,000 qubits, which would be a lot more realizable in the near-term hardware, which we could look at to implement the fast quantum algorithm in the future, maybe on the cloud or even within other industrial settings. And having requirements in mind for the depth of the quantum circuits and the corresponding compounds of interest can be investigated further in future generations of near-term hardware. And this is given by the fact that from the CellTalkDB repository that I had mentioned earlier in the presentation that I had looked at previously with the classical computing framework, it can be of much interest to determine whether there could be any near-term or any similar related compounds or similarly related families of compounds that could be examined on either current near-term quantum hardware or quantum hardware in the future. And in particular, determining, depending upon the error metrics that people at IBM, Google Quantum AI, or other quantum computing companies use, could actually be used to approximate better compounds that interact with a disease receptor of interest, which could have implications for either improved therapeutics or other types of biomedical or even more physics-oriented applications. So I wanted to provide an overview in which we discuss an overview or a broad level of fast quantum algorithms from the references that I provided in the first couple of slides of the presentation. And in doing this, we wanted to discuss connections with previous studies of variational quantum algorithms, which is a generalization of sorts of the variational quantum algorithm, as well as providing examples of compounds that have previously been studied with classical molecular docking workflows, which could potentially be also examined within quantum computing workflows so that we can establish a point of comparison between classical computation and quantum computation. And finally, we concluded by discussing prospects for being able to apply similar quantum algorithms like the fast quantum algorithm or the variational quantum algorithm or even the variational quantum eigensolver on future near-term hardware. And thanks for listening.